Hi there, welcome back. Have you ever wondered how our body utilizes oxygen so effectively? And how is it that we constantly have enough oxygen in our body until, of course, something goes wrong? Explained to a 5-year-old, hemoglobin is a bus that transports oxygen to their destination. Watch this video till the very end and see how it relates to hemoglobin binding and oxygen tension. This is a special series of oxygen transport where we will discuss oxygen content, hemoglobin, oxygen-hemoglobin dissociation curve. This is Mastering Anesthesia, so let's go.
Hi and welcome back. This is part 3 of oxygen transport. Today we are going to talk about oxyhemoglobin dissociation curve. So let's go. So what is oxyhemoglobin dissociation curve? It is a curve that describes the relationship between saturation of oxygen and blood oxygen tension. Before we dive into the details of the curve, let's get a few things clear. What are oxygen tension, saturation, SpO2 and SaO2? Are they the same? Are they interchangeable? If they're not, then what's the difference? So when we talk about blood oxygen tension, we are actually talking about the PaO₂. It is the pressure exerted by dissolved oxygen molecules in the blood. Okay, so the unit that we use is kilopascal or millimeters of mercury. And when we're talking about blood oxygen tension, it only reflects the oxygen dissolved in plasma, not the oxygen bound to hemoglobin. So if you remember the lecture before this, given in part of the oxygen transport series, we've talked about the percentage of oxygen dissolved in plasma is only about 2% out of the total oxygen content, but very, very important physiologically. Okay, the normal value for the blood oxygen tension is 80 to 100 millimeters of mercury, and this is for healthy adult breathing room air. Then what about oxygen saturation? We can either use SaO2 or SpO2. We'll get into the details later. It refers to the percentage of hemoglobin binding sites that are occupied by oxygen. So that is it, right? So how many of the binding sites of hemoglobin that is being bound by oxygen? The saturation refers to that. If it's 100% saturation, meaning that all binding sites have been occupied by oxygen. So if you use the bus analogy, the bus being the hemoglobin carrying for oxygen. So if it's fully occupied, meaning that saturation is 100%. But if it's only half occupied, so it is 50% saturation. The value of oxygen saturation can be obtained either by SaO2 or SpO2, but they are not one and the same. They have slight difference in its reading and how it's obtained. So for SaO2, it uses direct measurement of arterial blood. It measures how much the hemoglobin is saturated from the blood. So it is read from the ABG, and it is considered the true arterial saturation. SpO2, on the other hand, is measured peripherally. You can remember the P standing for peripheral. So it is the peripheral oxygen saturation. Measurement is by using pulse oximeter. The value is only estimated via light absorption through the tissues. And most stable patients, we can approximate the SpO2 as almost equal to SaO2. So SpO2 is not always equal to SaO2. It is not reliable in patients with shock, patient with hypoperfusion, where the SpO2 will not reflect the actual saturation of oxygen. Patients with nail polish also, we cannot get the SpO2, and severe anemia, and those with pathology in their hemoglobin. Let's take this understanding a step further. We want to look at how oxygen tension and oxygen saturation is being visualized through analogy. So if you take the same bus analogy, you have here 4 compartments, and this is at a bus stop. So the hemoglobin or the bus will carry 4 oxygens, and you have also oxygen around the bus, or passengers around the bus. You have a few here and a few there and a lot here. So the oxygen carried by the bus or the hemoglobin shows the oxygen saturation. Here you have 100% saturation, and those who are waiting at the bus stop this causes tension. So this the oxygen surrounding the bus, outside the bus, this is oxygen tension, and the total of the oxygen saturation and oxygen tension surrounding the bus, this contributes to the total oxygen content in the blood. Now let's talk about the curve of the oxyhemoglobin dissociation curve. The curve has a sigmoid shape. This is due to the features of hemoglobin which have cooperative binding. If you have not watched part 1 of this oxygen transport, I suggest you do so. You can find the link down below in the description. Let's talk about the clinical consequences of oxyhemoglobin dissociation curve having this sigmoid shape. You can see the consequences from the curve having this steep portion and this flat plateau portion. So what happens at the upper portion of the oxygen hemoglobin dissociation curve? It is flat. So what happens is that even if the patient's PO2 falls, the saturation barely change. Let's plot this out on the graph to visualize the idea. Let's take a drop of oxygen tension from, let's say, 13 to 10 kilopascal, and reflecting on the saturation of oxygen, you can see that the drop is very minimal from, let's say, 100 to it's about 95. So that demonstrates that drop in oxygen tension, or the flat upper part of the curve, causes only a slight reduction of saturation of oxygen. Now let's see how lung pathology plays a role in this ODC curve. So it is dangerous in patients with lung pathology or any state with low PaO2. They will be on the steeper part of the curve. So let's take, for example, a person with a lung pathology that has brought down the oxygen tension to only about 5 kilopascal, right? So if the partial pressure drops even a little, let's say from 5 to 3.5 kilopascal, the drop in saturation of oxygen will be massive. You can see there's this large desaturation that occurs. In normal situation, the steep part of the curve reflects what is happening at the peripheral tissue, where the oxygen tension is basically low. And as you can see, large amounts of oxygen is offloaded just for a fraction of drop in oxygen tension. You can see here the drop from 3.5 to 3 kilopascal causes this huge offloading of oxygen, where the drop is from 40 to 30%, meaning that the oxygen has been offloaded from the hemoglobin. Let's take a closer look at the oxyhemoglobin dissociation curve. You have saturation of oxygen against oxygen tension in kilopascal, percentage for the saturation of oxygen, and kilopascal for oxygen tension. You can also write it in millimeters of mercury. You have to multiply the numbers with 7.5. So at 13.3 kilopascal, it's approximately 100 millimeters of mercury. And at 10 kilopascal, it is about 75 millimeters of mercury. So what can be derived from this oxyhemoglobin dissociation curve? One of the most important thing that we can get from oxyhemoglobin dissociation curve is the P50 value, which is to describe the position of the oxyhemoglobin dissociation curve. The P50 is the partial pressure of oxygen at which 50% of hemoglobin is bound to oxygen. In other words, at P50 value, it is a point where 50% of the hemoglobin is occupied by oxygen, and the other half of oxygen has dissolved in plasma, meaning that they have been offloaded from the hemoglobin and dissolved in plasma. It is the midpoint of the oxyhemoglobin dissociation curve. So if the P50 shifts slightly to the right, it means that the ODC curve has shifted to the right. If the P50 value is lesser than the normal value, meaning that the oxyhemoglobin dissociation curve has been shifted to the left. So let's see this on the graph, shall we? So at saturation of 50%, we trace it to the oxyhemoglobin dissociation curve. Continuing the tracing, we find out that at 50% saturation of hemoglobin, the oxygen tension is found to be at 3.5 kilopascal. Convert this to millimeters of mercury. This is equivalent to 26 millimeters of mercury. So P50 value is 26. Normal range given by many books say that the normal value is about 24 to 28. For ease of discussion in this topic, I will use P50 value as normal as 26. So if we check on the ABG and found out that our P50 value is larger than 26 to 28, means that the oxygen hemoglobin dissociation curve has shifted to the right. It means that the affinity of oxygen for hemoglobin has reduced, causing the oxygen to be easily offloaded from the hemoglobin into the blood and into the tissues. We can see that at saturation of 50%, we found out that the oxygen tension is higher, meaning that much of the oxygen has been offloaded by the hemoglobin and dissolved into the plasma. Then, if our ABG shows a value of less than 26 or 24, it shows that our oxygen hemoglobin dissociation curve has shifted to the left, meaning that our hemoglobin has increased in oxygen affinity. Just to demonstrate further, let's look at this graph that has shifted to the right. We found out that at saturation of 50%, the PaO₂ oxygen tension is at 4 kilopascal, or about 30 millimeters of mercury. So since P50 is more than 26, we have concluded that this graph has shifted to the right. And on the other hand, we see that at saturation of 50%, the graph has a oxygen tension of only 3 kilopascal, or 22 millimeters of mercury. So since the P50 value is less than 26, we can conclude that this graph has shifted to the left. So what causes the shift of the oxyhemoglobin dissociation curve to the right? Remember the mnemonic cadets, where we have C for increase in carbon dioxide, acidosis, where there is increase in hydrogen ion, 2,3-DPG, or diphosphoglycerate, increase in the level of 2,3-DPG, E for exercise, T for increase in temperature, and the presence of HbS, sickle cell hemoglobin, present in sickle cell disease. Next, we know the oxyhemoglobin dissociation curve shifts to the right, but what are the underlying physiological mechanisms behind this rightward shift? The most famous mechanism related to this rightward shift is the Bohr effect. It is when metabolically active tissues causes an increase in local carbon dioxide, heat, and hydrogen ions, causing the local site to be more acidic. So the blood arrives at the capillaries of this tissue. The oxygen hemoglobin dissociation curve shifts to the right at this site. So at this point, there will be increase in oxygen offloading from the hemoglobin because the tissue needs it, right? So in simple terms, carbon dioxide, heat, and acidosis tells hemoglobin to drop off oxygen. That is it. That is all about Bohr effect. There's nothing complicated about this. Take this into the bus analogy. You have the hemoglobin carrying oxygen to a site, this local tissue site, saying Bohr. So when all the conditions are met, the Bohr effect causes the oxygen to be offloaded from the hemoglobin. The second mechanism responsible in this rightward shift is the anaerobic metabolism. So it happens when the PaO₂ level at the cellular level drops below a critical threshold value, known as Pasteur point, in which less than 1 millimeters of mercury at the mitochondrial level, the metabolism will convert itself to anaerobic metabolism, right? The anaerobic metabolism will predominate. In anaerobic metabolism, glucose undergoing several pathways will be turned into lactate. Along the way, energy will be produced. On a side pathway in anaerobic metabolism, there will be also a production of 2,3-DPG. Okay, so 2,3-DPG is directly proportional to the level of anaerobic metabolism in the body. So as the level of anaerobic metabolism in the body gets more active, so does the level of 2,3-DPG in red blood cell. So remember from our previous lesson, 2,3-DPG binds to the beta subunit of hemoglobin, stabilizing the configuration, making hemoglobin difficult to bind to oxygen. So this results in decreased affinity of oxygen for hemoglobin. Next, let's see what happens in the lung. There will be oxygen loading in the lungs. So you have red blood cell in the blood. We have alveoli in the lungs. Okay, so when the blood reaches the alveoli, carbon dioxide will diffuse out from the blood into the alveoli. This will normalizes the pH, causing the P50 to return to its central position. So oxygen affinity will increase. There you can see oxygen binds to the hemoglobin, goes back into the hemoglobin. So when levels of oxygen in the blood reduces, this will cause oxygen from the alveoli to be absorbed from the capillaries into the blood and subsequently into the hemoglobin. Now let's talk about oxygen hemoglobin dissociation curve shifting to the left. So what causes leftward shift of oxyhemoglobin dissociation curve? Remember our cadets? Now exactly reverse that. So conditions like reduction in SaO2 level, alkalosis, reduced 2,3-DPG in the red blood cells, and hypothermia all will contribute to the leftward shift of oxyhemoglobin dissociation curve. Apart from that, other hemoglobin pathology also contributes to leftward shift of oxyhemoglobin dissociation curve, such as carboxyhemoglobin, methemoglobin, and also fetal hemoglobin or HbF. Do not get confused with HbS of sickle cell, because in that case, the oxyhemoglobin dissociation curve will shift to the right. But carboxyhemoglobin, methemoglobin, as well as fetal hemoglobin will cause the ODC curve to shift to the left, increasing oxygen affinity towards hemoglobin and reducing oxygen offloading to the tissues. In leftward shift, the hemoglobin's affinity towards oxygen will be increased. This is particularly important physiologically in the fetal life. In fetus, they need to extract oxygen from maternal blood. The hemoglobin affinity needs to be higher than hemoglobin oxygen affinity in the mother's blood to extract the oxygen from the maternal side to their side of the blood through the placenta. There is, however, another problem. Red blood cell in the fetus also have 2,3-DPG. Will this cause a problem? No, because 2,3-DPG only binds to beta globin chain of hemoglobin, whereas HbF has gamma globin chain, so 2,3-DPG cannot bind to the hemoglobin, and this further enhances the affinity of HbF towards oxygen. And there you have it. Thank you so much for watching my videos. I hope you have benefited something from these videos. Please like, comment, share, subscribe. If you have any corrections, suggestions, please comment down below. And I'll see you in my next video.
Thank you for watching. If you find these videos helpful, please hit the like and subscribe button to show your support. Comment down below what topic you want us to cover next. Till next time, see you in the next video.